Yesterday, I was reading through a lab report and came across this unsettling detail. It wasn't about a new molecule or a surgical technique, but about a scientist successfully taking a mouse's specific fear memory, say, the memory of a minor electric shock, and with a few chemical tweaks, effectively switching it off. No trauma therapy, no reliving the moment, just a precise epigenetic edit to the DNA's packaging inside a few neurons. What was interesting was the speed of the shift in the field. Between 2023 and 2025, it felt like the entire biomedical world raced from theory to application. We went from talking about genetic risk factors for Alzheimer's to researchers using CRISPR to convert the high-risk APOE4 allele in a petri dish, leading to a massive drop in the pathological tau protein that clogs up the brain. The consensus was one of giddy excitement. The long-awaited key to logging away PTSD, curing addiction, and halting dementia was finally here. But then I reached the final section of the report, the part discussing the ethical challenges. Just as that initial excitement about the therapeutic potential was cresting, the whole tone changed. Suddenly, the focus wasn't on healing, but on a strange, profound warning. The ability to edit memory didn't just promise to remove pain. It introduced the possibility of what the researchers call self-alienation, where a perfectly treated person feels fundamentally disconnected from their own life story. The whole narrative identity they had built, their values, their motivations, all suddenly felt like a lie told by a stranger. That instantaneous pivot from the exhilarating promise of a cure for human suffering to the terrifying prospect of losing the very self that suffers, it honestly left me kind of stunned. It made me feel that this memory editing revolution is less about conquering disease and more about a much deeper, more consequential question. What is the price we pay for escaping our past? And who gets to decide which memories are worth keeping?
Okay, yeah, let's unpack this. That introduction, it perfectly encapsulates the fundamental challenge we face right now.
It's a huge one.
It is. We have this rapidly advancing biomedical solution, a molecular key, if you will, to some of the most profound human suffering, things rooted in memory and trauma.
Right.
But using that solution, actually applying it, requires us to redefine the very concept of the self. So for this deep dive, we absolutely have to balance the exhilarating promise of the science, which is moving at a truly breakneck pace, with this profound, complex need for ethical governance and foresight.
That pivot you mentioned, from fixing cells in a dish to this existential crisis, it's jarring.
Mm-hmm.
And the speed of that technological shift is really the first thing that just jumps out. In just a couple of years, the entire field went from theoretical possibility to serious preclinical and clinical application at lightning speed.
The acceleration is undeniable. It's really been driven by the refinement of two major technological paths that have sort of emerged. The first one is epigenetic editing. It's often described as the reversible switch. Okay. This came out of these landmark discoveries, particularly by scientists at EPFL, who figured out how to influence memory circuits without actually altering the genetic code itself.
I really like that analogy someone used, that it's about changing the settings, not the words in the book.
Yeah.
It's making the DNA easier or harder for the cell to actually read.
Exactly. We're talking about modifying the packaging of the DNA within the neurons. This is done through what we call chromatin remodeling machinery. So, like you said, we're not changing the fundamental DNA sequence.
Right.
But we are modifying how genes are accessed and expressed. For memory, this means using enzymes to add or remove chemical tags, things like acetyl groups or methyl groups, on the histone proteins that the DNA wraps around.
So, if a memory is stored in a neuronal circuit, and that memory is, say, way too strong, like in PTSD, epigenetic editing could potentially loosen the grip of the DNA on those histone proteins.
Precisely. If we want to strengthen a memory association, we might use something called a histone acetyltransferase to open the chromatin up, making the memory-related genes highly accessible for expression.
And to weaken one.
If we want to weaken a traumatic memory, we might encourage histone demethylation. This non-permanent reversible aspect is a huge advantage. It lets you fine-tune specific memory associations without the permanent commitment of a full genomic edit.
And that brings us to the second, more durable path, which is using CRISPR-Cas9 for tackling these really severe genetic roots, specifically for devastating diseases like Alzheimer's.
That's where the profound therapeutic promise really lies. CRISPR is delivering some incredibly compelling preclinical results by directly correcting inherited risk factors. Right. The prime example here is the high-risk APOE4 allele,
which is a major, often definitive genetic factor for late-onset Alzheimer's.
It is. So researchers have used CRISPR to convert that high-risk high-risk allele into the neutral APOE3 version, and they did this in patient-derived stem cells.
So what was the mechanism for the correction they saw? What exactly happened to the cellular pathology when they made that change?
The pathological correction was, well, it was dramatic. You have to remember, Alzheimer's is largely driven by the accumulation of misfolded proteins, specifically tau and amyloid beta, or APAX.
Yep.
Plaques and tangles.
Exactly. The corrected cells, these were neurons derived from patient stem cells, they showed a massive drop in the hyperphosphorylation of that pathological tau protein. And on top of that, their A42 secretion levels just plummeted.
So by correcting a single genetic letter, they effectively stopped the cellular cascade that leads to all that junk clogging up the brain.
They essentially restored the cellular health to a state that's comparable to healthy neurons that only have the APOE3 allele. This is monumental.
Yes, sounds it.
It suggests that correcting the gene could halt the fundamental disease process rather than just, managing symptoms. But, and this is a huge caveat.
I was waiting for the but. This is where the caution comes in. While the correction is demonstrated beautifully in a Petri dish, safely delivering the CRISPR machinery across the blood-brain barrier and into enough neurons in a living human brain, that remains the primary, highly complex engineering bottleneck.
And outside of just addressing a genetic predisposition, we're also seeing work focused purely on neurological repair and restoration.
Absolutely. Moving past symptom management toward actual tissue preservation, a key neurorestorative strategy that's currently in advanced clinical testing for early Alzheimer's involves gene therapy using AAV2-BDNF.
Okay, so BDNF is brain-derived neurotrophic factor. For listeners who might know the name but not the function, what does BDNF actually do in the brain, and how does this gene therapy use it?
BDNF is often described as, fertilizer for the brain. It's essential for the survival, growth, and differentiation of neurons.
So it helps them stay healthy and grow.
Yes. And this therapy uses an adeno-associated virus, or AAV2, as a vector to deliver the BDNF gene into brain cells. Once it's delivered, those cells continuously produce the BDNF protein. The whole approach aims to prevent the progressive neuronal loss and slow down cognitive decline by activating something called the TRPB receptor on target neurons.
So it's not trying to edit the memory itself or even correct a genetic fault. It's designed to protect the existing infrastructure and keep the brain functional for longer. It's like a proactive defense against decay.
It marks a critical shift toward restorative interventions. We're trying to leverage the body's own protective mechanisms to achieve long-term functional preservation and plasticity. It highlights the breadth of tools we now have available, with each one targeting a different point in the pathology.
Let's put these tools side by side then. We've got the instant switch, the durable edit, and this neurofertilizer. What is the fundamental mechanical trade-off we're making when choosing one of these tools? Is it always a case of precision versus durability?
That is the core calculus in neurotechnology right now. Millisecond precision often comes with high invasiveness, whereas, durable correction demands complex delivery and carries the risk of unintended permanence.
Let's start with optogenetics. This is the one that gives researchers instantaneous, real-time control over neural circuits. Right.
Correct. This method uses viral vectors to deliver light-sensitive proteins, called opsins, into specific populations of neurons. So, for example, channel rhodopsin 2, or CHR2, is a common activating opsin, and Hiller-rhodopsin is an inhibitory one.
So, depending on which opsin you deliver, you can either turn a targeted neuron on or turn it off just by shining a specific wavelength of light on it.
Exactly. When you expose them to light, and this is typically via invasive fiber optics inserted into the brain, these opsins act as instant high-speed switches.
Wow.
This gives you millisecond precision, which is just phenomenal for studying memory formation or understanding how specific circuits function, or even immediately suppressing acute traumatic associations like a specific PTSD flashback being triggered.
But I've heard the clinical drawbacks are massive.
Yeah.
I mean, how realistic is optogenetics for widespread therapeutic use when it requires invasive brain surgery just to shine a tiny light?
That is the major limiting factor for clinical scalability. It requires invasive brain implants to deliver that light signal. And furthermore, while the timing is incredibly fast, some of these opsins can stay active for up to 156 milliseconds.
Which is an eternity in brain time.
It is. In the fast-paced electrical environment of the brain, that risks what we call non-physiological overstimulation. This prolonged activity could potentially disrupt normal network function and could even trigger seizures, which is a substantial safety challenge.
Which brings us back to the durable, long-term modulation approach, the CRISPR epigenetic editing. This one sacrifices speed for stability and, crucially, potential reversibility.
Precisely. This aims for a durable, sustained change. It doesn't use the CRISPR enzyme Cas9 to actually cut the DNA. Instead, it uses a deactivated CRISPR system, dCas9, which acts as a guide. It latches onto a specific DNA sequence without cleaving it.
And what does it bring along with it? What's the payload?
It's fused to epigenetic enzymes, and that's the key. For memory manipulation, these enzymes could be histone acetyltransferases, or HATs, or histone demethylases. These are the workers that add or remove those chemical tags on the DNA packaging proteins, effectively turning a gene's expression level up or down.
I see.
This sustained modulation is ideal for chronic, long-term disorders like Alzheimer's or addiction, where you need a fundamental, steady state change.
That sounds incredibly precise. But the risk of off-target effects, or what you could call genomic shrapnel, that still looms large, doesn't it? Especially because this change is meant to be durable.
That is the paramount safety risk for any kind of durable gene editing. Even with optimized guide RNAs, the system can mistakenly recognize and bind to similar-looking DNA sequences elsewhere in the genome.
How much wiggle room is there?
The guide RNA tolerates up to 3 DNA mismatches. That's the tolerance level. This means the risk of unintended genomic edits, disrupting critical non-targeted genes, is always present. For chronic disorders where the therapy is designed to persist for decades, mitigating these off-target effects through highly refined vector design and delivery is absolutely essential.
So the science of editing the memory is ready, but the engineering challenge of accessing the memory is huge. As we said earlier, the brain is the body's fortress. Before any of this works, we have to figure out how to bypass those physical defenses. For CNS gene therapy, that's arguably the single biggest technical hurdle.
Oh, the complexity of accessing the central nervous system, the CNS, is staggering. It's not just a single wall. It's a multi-layered defense system designed by evolution to keep almost everything out.
Right.
And here we are trying to deliver these large, complex payloads, AAV vectors, nanoparticles, even whole CRISPR systems to specific tiny populations of neurons deep within the brain.
Let's detail those specific barriers. The most famous one, of course, is the blood-brain barrier, the BBB.
The BBB, and its counterpart, the blood-spinal cord barrier, or BSCD, is incredibly restrictive. It prevents over 99% of systemically injected drugs from ever entering the brain parenchyma.
Wow, 99%.
Structurally, it's composed of these highly specialized endothelial cells that are linked by tight junctions, and those cells are further surrounded by pericytes and the end feet of astrocytes, forming this neurovascular unit that strictly controls all molecular traffic. It's a filtration system based on size, lipid solubility, and electrical charge.
And even if a therapeutic agent manages to sneak past that endothelial wall, it still faces the crowded environment inside the brain tissue itself.
That's the second line of defense, the extracellular space, or ECS. Yeah. This space, which is the fluid-filled matrix between brain cells, acts as a secondary variable diffusion barrier within the brain. It's not a simple bath.
It's not just empty space.
Not at all. Its geometric structure, its tortuosity and volume fraction acts like a sieve limiting movement.
So large viral vectors or nanoparticles have to physically worm their way through this crowded soup of molecules and cells.
Precisely. The ECS significantly influences vector uptake, metabolism, and clearance dynamics. On top of that, glial cells, particularly astrocytes, are highly active in the ECS and can modulate drug effects and distribution. And the characteristics of the ECS also vary from one brain region to another, which just complicates achieving a uniform distribution for any treatment. It's a logistical nightmare.
This is where engineering has to meet biology in the most innovative way possible. How are researchers solving this seemingly intractable problem of getting the goods to the exact right address in the brain?
This challenge has pushed innovation into the realm of, well, science fiction, frankly, and it underscores the high-stakes technical reality of this field. I remember reading an online article that was discussing the initial clinical trials of advanced neurodelivery systems. This was in a specialized research hospital setting. The focus was on getting that AAV2 BDNF gene therapy to combat early-stage Alzheimer's.
What was the specific engineering solution they developed to get that kind of hyper-precision?
They described the painstaking work of a lead engineer, a Dr. Chen. His team developed a truly tiny, biodegradable micro-robot system. These aren't just passive carriers. They're actively guided by external magnetic fields to ferry the gene-carrying vectors across the blood-brain barrier and into the targeted neurons like those in the hippocampus with unprecedented precision.
A microscopic surgical strike force for the mind.
That's exactly the term Dr. Chen used. He said, and I'm paraphrasing here, that they had built a surgical strike force for the mind. But he quickly followed that up by noting the incredible weight of responsibility. He said, every tiny adjustment, every navigational error carries the weight of a person's entire memory.
Wow.
It just highlights the terrifyingly high-stakes level of technical precision that's required. Because a distribution error doesn't just waste the drug, it could affect unintended critical neural networks.
Let's break down the modalities being used. We have the two broad categories: direct, invasive delivery, which is more established, and then the remote-controlled, non-invasive methods.
Starting with the invasive methods, intrathecal administration, or IT, is a major pathway. This involves injecting the therapeutic agent directly into the spinal canal.
Which means using the cerebrospinal fluid, the CSF, as the transport medium.
Yes. The CSF acts as a conduit, and it facilitates the distribution of the genetic material throughout the entire CNS. While it's less invasive than directly piercing the brain, distribution via the CSF isn't always uniform, which limits its utility for targeting specific deep brain structures.
So it's better for diseases that are more spread out.
Exactly. It's often best for diseases where the pathology is spread across the CNS, like spinal muscular atrophy or ALS.
The other major invasive method is highly precise, focusing on getting high concentrations to a specific site. That's convection-enhanced delivery, or CED.
CED is essentially a direct infusion. It bypasses the systemic circulation and the BBB entirely by placing the agent right into the target tissue. It's a method that has moved robustly into clinical trials for disorders like Parkinson's, using AAV2-GDNF.
The results from the CED trials for something called AADC deficiency were particularly striking. They really demonstrated the power of precise target delivery.
They were revolutionary. AADC deficiency is a devastating pediatric disorder that affects motor function. In these children, the direct bilateral infusion of AAV2 AADC into the midbrain proved safe and well tolerated. The goal was to target the substantia nigra, and the precision they achieved was incredible. Up to 98% of the target tissue was covered.
And what did that high coverage translate to clinically for these children?
The clinical improvements were profound and life-altering. The involuntary eye movements, known as oculogeric crisis, were completely resolved in 6 out of 7 patients within 3 months. And 12 months post-surgery, 6 out of 7 children gained normal head control, and critically, 4 of them could sit without assistance.
Think about that for a moment. This isn't just symptom mitigation. This is restoring fundamental motor control and developmental milestones via a single, precise infusion. That just underscores why researchers are willing to tackle the complexity of invasive delivery.
It demonstrates the profound feasibility and clinical payoff of targeted midbrain gene delivery via CED. It shows that if you can deliver the correct payload to the correct place, the brain has an enormous capacity for repair and functional restoration.
Now let's pivot to the non-invasive side. The ways we try to temporarily compromise the BBB so we can deliver systemically injected therapeutics. Focused ultrasound, or FUS, seems to be the star here.
FUS is really the cutting edge of non-invasive delivery. It combines targeted ultrasound waves with... Exactly. And it's been used successfully in macaque monkeys and now in patients. Clinically, FUS has facilitated the uptake of molecular tracers like 18F-choline. Crucially, in Alzheimer's patients, FUS-mediated delivery of anti-amyloid antibodies, like aducanumab, significantly reduced cerebral amyloid beta load.
And that reduction was localized only to the areas where the ultrasound was applied, demonstrating its precision.
Yes, the reduction was markedly greater in the FUS-treated regions than in untreated areas, confirming its targeting efficacy. What's more, FUS has been applied for transiently opening the BBB to allow systemically delivered CRISPR-Cas9 machinery to reach the brain.
So FUS is an essential enabling tool for the long-term goal of non-invasive memory editing.
It absolutely is.
Beyond viral vectors, what about the alternative non-viral vehicles? If we don't want the risk of a viral immune response, what are researchers working on?
Vector innovation is crucial here. 2 major non-viral alternatives are polymeric nanoparticles, or PNPs, and exosomes. PNPs are synthetic carriers that can be surface modified to target specific receptors on the BBB, which enhances their penetration.
And exosomes, those naturally occurring cell vesicles, They seem promising because of their inherent biocompatibility.
Exosomes are fascinating. They are naturally occurring extracellular vesicles that cells use to communicate with each other. They offer an inherently biocompatible, biodegradable, and non-immunogenic platform.
So the body doesn't see them as a threat.
Right. They demonstrate robust stability and a proven promise for BBB penetration. Furthermore, if you use exosomes derived from the patient's own cells' autologous exosomes, you further mitigate the risk of immune reactions.
But if they're natural, do they have limitations on how much genetic payload they can carry?
They do. They're limited by their inherent cargo capacity, which is a significant drawback when you're trying to deliver large complexes like a full CRISPR system. There are also substantial challenges in scaling up their production and purification for widespread clinical use. So while they're very biocompatible, they face significant manufacturing and cargo restrictions that PNPs and AAV vectors currently manage better.
So even with these brilliant delivery systems, the regulatory path is incredibly complex. If you are administering a gene therapy product into a human brain, especially via invasive methods like an intrathecal injection, the FDA standards must be absolutely stringent. What does the agency focus on to ensure safety?
The FDA's guidance for gene therapy products for neurodegenerative diseases details extremely stringent requirements under what's called Chemistry, Manufacturing, and Controls, or CMC. Their absolute focus is on product purity. This requires rigorous testing for any product-related impurities.
What kind of impurities are we talking about? Empty viral shells?
Exactly. We need to measure and minimize empty viral particles, partially filled particles, and most critically, the presence of replication-competent viruses, or RCVs. An RCV is a full viral particle that could reproduce uncontrollably, which is an unacceptable risk in the CNS.
And what about contaminants from the manufacturing process itself? Things left over from the culture media?
Those are process-related impurities, and they are subject to very strict limits. This includes residual host cell proteins and, most critically, residual host cell DNA. This is a major concern specifically in neuronal tissue because neurons have low cellular turnover. They don't divide rapidly.
So if residual DNA, which might contain potentially tumorigenic sequences, gets into a non-dividing neuron, it might just sit there indefinitely, increasing the theoretical risk of cancer.
Precisely. Unlike quickly dividing tissues where residual DNA might be diluted out, administered host cell DNA impurity may persist for a prolonged period in the brain. The FDA recommends manufacturers carefully consider the characteristics of the cell lines they use to ensure they don't contain tumorigenic sequences and minimize residual DNA levels to the lowest possible limit of detection.
I also noted the very specific requirements for intrathecal administration, where even tiny contaminants can be a huge problem.
Given the direct route to the CNS via the spinal canal, the tolerance for contaminants is extremely low. Specifically, endotoxin levels, which are components of bacterial cell walls, must be kept below an extremely tight threshold of 0.2 endotoxin units per kilogram of body weight per hour.
Because an inflammatory reaction in the spinal fluid would be a catastrophe.
It would be catastrophic. A systemic reaction is bad enough, but one directly in the CSF is something you have to avoid at all costs.
It sounds like the FDA isn't just looking at the chemical makeup, but also anticipating the design risks unique to these gene therapies.
They're looking at the whole risk matrix. Gene therapy products have to be designed to mitigate critical risks, like reducing unintended immune responses, which can compromise treatment efficacy and cause inflammation. They must also minimize the possibility of the viral vector becoming latent in neuronal tissues and then reactivating later on.
Right.
They even recommend against including foreign genes that lack biological function for the therapy, like generic reporter genes, just to simplify the risk profile.
And finally, given the complexity of the delivery systems we just discussed, the specialized pumps, the CED cannulas, or even those theoretical microrobots, how is that device compatibility regulated?
Device compatibility is a non-negotiable requirement. It has to be demonstrated before initiating phase 1 safety studies. The sponsor has to demonstrate that the investigational product, the gene therapy, is fully compatible with the specific delivery device.
What does that involve?
It involves testing to ensure there is no loss of strength or potency or increase in impurities when the product is passed through the device under conditions that exactly mimic the intended clinical study. If the device is used outside its clear indications, early regulatory discussion is mandatory to inform the safety evaluation of the combined product-device system.
Okay, this is where we shift completely. We're moving from the microscopic battle inside the CNS to the macroscopic question of what it means to be human. We've achieved this capability to eliminate suffering, but the person who endured the struggle and whose life story was partially defined by that endurance, they might cease to exist as they knew themselves.
That feeling of tension between therapeutic relief and loss of identity is the crux of the profound warning the researchers cited. Let's ground this with a concrete example. I recall a story about a woman named Anya who was paralyzed by the crushing weight of post-traumatic stress disorder following a severe accident.
And what happened after she received the epigenetic therapy?
Well, the treatment was a success. She remembered the accident, but the paralyzing visceral fear was gone. She could finally breathe. But the first time she tried to tell the story of her recovery to a friend, she hesitated.
What did she say?
She whispered that it felt like, I'm remembering a book I once read, not something that happened to me. The memory was factual, but the emotional truth had just evaporated.
That subtle shift is devastating. That feeling of disconnect where the memory is factual but totally inert is the core concept of self-alienation.
Yeah.
The healing is undeniable, yet this shadow remains.
Yeah.
Is the edited self the true self?
It creates a profound disruption to what philosophers call our narrative identity. Memories are not just recordings of events. They are the fundamental building blocks of the self-narrative we construct over time. When you alter the significant emotional or first-personal aspects of a memory, you risk the individual feeling fundamentally disconnected from their entire life story, their values, and their long-term motivations.
Help us unpack that difference between mere episodic memory and narrative identity. If Anya can still recall the facts of the accident, why does she feel alienated?
Episodic memory is the factual recall, what happened, when, and where. Narrative identity, however, is about the coherence of the self. Who am I, and why did I make the choices I made? The integrity of the first personal aspects of memories, that feeling that I suffered this and I grew from it, is crucial.
And Anya's memory became third person.
Exactly. The emotional valence that connected the event to her present self was gone.
And this erosion of emotional responsiveness can affect so much more than just personal history. It can affect one's moral sensibility, too.
Absolutely. Our capacity to feel emotions like blame, regret, or even guilt are intimately tied to the emotional content of past experiences. If memory modification treatment, or MMT, systematically dampens that emotional responsiveness, it risks eroding the very capacity to feel those core moral emotions that guide our future actions.
The Claude case offers an even more powerful, tangible example of how removing a painful memory can tragically dismantle a positive, defining life purpose. Tell us about Claude.
Claude had suffered deep psychological trauma in his childhood. That trauma was the primary visceral motivation for his career choice. He founded and dedicated his life to a significant local charity supporting abuse victims. He underwent MMT to relieve his paralyzing psychological burden.
And it worked.
It did. The MMT was successful, his fear and distress were gone.
But the therapeutic success led to an existential vacuum.
It did. Because the dedication driving memory was no longer grounded in the same visceral, emotional reality, Claude experienced a profound shift. He started contemplating abandoning his charity. The passion, the urgent need he felt to serve, it vanished because the emotional content that provided the reason for his sacrifice was absent.
And this is defined as a loss of authenticity because his most important life choice, his philanthropic career, would no longer be grounded in his long-standing defining values born from his trauma.
That is the philosophical danger. The treatment decreased Claude's narrative coherence. He was experiencing self-alienation because he could no longer make practical, evaluative sense of his past choices. The lack of emotional content meant he lacked the moral reasons for or against certain avenues of action. He knew he ran the charity, but he couldn't grasp why it mattered to him anymore. His authentic self was lost in the process of healing.
And furthermore, if memory modification lessens the deep, empathetic connection to suffering, as it did for Claude, it threatens social advocacy and political action.
That's the critical societal dimension. Blunting the emotional component of memories carries the undeniable risk of lessening empathy and advocacy for social change. Many movements for justice are founded on the profound emotional memory of past inequities or violence.
Right.
If MMT is applied broadly, it could hinder the collective emotional drive needed to lessen those inequities. Moreover, the political risk is clear. Authoritarian regimes could weaponize this technology to erase collective memories of state violence or oppression, effectively rewriting citizens' personal histories and suppressing dissent.
The disruption doesn't stop at personal identity or political stability. It touches the foundations of justice itself. What are the immediate legal conflicts we face if memory editing becomes widespread?
The ability to alter recollections directly compromises the reliability of personal testimony, which is the absolute cornerstone of our legal processes. This is especially true if a witness or even a victim undergoes MMT.
And this question of integrity is immediate, even with existing less invasive drugs, like the beta blocker propranolol, which has to be administered right after a traumatic event to dampen memory consolidation.
That immediate window creates a massive legal and ethical challenge of obtaining adequate informed consent. How can a recently traumatized patient, often in a state of shock, possibly know the full long-term consequences of choosing to chemically damp down a defining memory?
They can't. They can't know if they will later find meaning in that experience or redeem those painful memories. There's an enormous uncertainty about the cost of losing a component of their self-identity versus the immediate therapeutic benefit.
And for the judicial system, memory dampening on a bystander witness creates an immediate trade-off. We are balancing the individual's autonomy to heal versus society's interest in preserving evidence that's vital for prosecution. If memory dampening affects factual recall, it could even be deemed the spoliation of evidence.
Wow.
We would then be forced to create a legal defense based on medical necessity versus the obstruction of justice.
Yeah. And then there's the economic and legal pressure in civil court, the problem of mitigation of damages.
In civil tort law, a plaintiff sues to recover damages, often including compensation for pain and suffering and emotional distress. If MMT becomes highly effective and standard practice, courts could begin pressuring plaintiffs to mitigate those emotional distress damages by choosing to dampen their memories,
Effectively penalizing those who refuse the treatment based on their deeply held interest in retaking their full experience. They might say, why should we pay you for suffering you refuse to eliminate?
Exactly. It creates a coercive environment where personal values conflict with legal and economic pressure. This is a profound interference with personal liberty disguised as therapeutic rationality.
Finally, let's address the elephant in the room: socioeconomic inequality. This technology is brilliant, it's complex, and it is hugely expensive to develop and deliver. That threatens to make MMT a luxury item for the privileged few.
This is perhaps the most urgent social crisis this technology presents. I heard a poignant anecdote from a colleague who attended an international policy conference in Geneva in 2025, and it was focused on equitable access to neurotechnology.
Well, I haven't at the conference.
A young delegate from a country with a fragile, underfunded healthcare system stood up and spoke of the terrifying risk of creating two distinct human races. One whose suffering could be edited away and whose psychological well-being was chemically assured, and another whose pain was permanent, leading to perpetual mental and emotional disparity.
That's the ultimate social disparity. A divide not just in access to physical health, but in fundamental mental and emotional stability.
The high cost of development and specialized delivery means MMT threatens to become a luxury enhancement, not a universal treatment, thereby exacerbating existing social stratification. Unequal access would create a profound ethical and social chasm. Policy has to proactively address this, developing specific equity plans to ensure these therapies are accessible and affordable, or we risk a world where trauma is an inherited condition of the poor.
So the need for robust, adaptive, and trustworthy governance is crystal clear, given the dual risks of technical failure and profound ethical misuse. What are the major policy bodies doing to manage this balance between the speed of innovation and the safety of society?
Regulatory bodies like the FDA are adapting rapidly. For advanced, highly technical treatments, the FDA approach utilizes expedited programs like the Regenerative Medicine Advanced Therapy designation, or RMAT. This accelerates the development and review of therapies intended for serious conditions with unmet medical needs, which neurodegenerative diseases certainly qualify as.
But accelerated review cannot mean compromised safety standards, especially when you're dealing with durable genetic changes in the brain.
Absolutely not. The FDA strongly emphasizes the need for robust clinical trial design. They recommend randomized, concurrent controlled trials whenever possible, even for first-in-human studies.
Why is that so important?
It's crucial because these gene therapies have sustained biological activity over a person's lifetime and often preclude repeat administration because of immune responses. You get one shot to prove safety and effectiveness efficiently. They also mandate continuous early communication through mechanisms like Interact meetings, long before the formal submission of an investigational new drug application, or IND.
Moving beyond national regulation, what is the international standard attempting to govern this complex philosophical landscape of neurotechnology?
The guiding light here is the OECD recommendation on responsible innovation in neurotechnology, which was adopted back in 2019. This is the first international standard in this domain, and it aims to guide governments and innovators globally to anticipate and address the ethical, legal, and social challenges raised by these powerful new tools.
And what are the key tenets the OECD outlines to protect the individual in this new technological era?
The principles are heavily focused on safeguarding core human rights. They specifically promote the principles of cognitive liberty, which is defined as the right to mental self-determination, and the individual's autonomy over their own mind and brain processes.
I see.
They also mandate safeguarding personal brain data from being used in discrimination, particularly in employment or insurance contexts, where an insurer might demand to see your brain data before offering a policy.
And given the unpredictable, rapid nature of neuroediting, what steps are required to make sure these oversight bodies aren't always just playing catch-up?
The OECD mandates that oversight bodies must build the capacity to conduct horizon scanning and scenario planning. This isn't just passive observation, it's an active process to anticipate potential unintended uses and, deliberate misuse.
So what would that look like in practice?
Well, for example, scenario planning would look at how an MMT tool designed for PTSD could be repurposed to influence democratic decision processes by purposely affecting freedom and self-determination on a mass scale. They must also promote cultures of stewardship and trust, demanding transparent communication that rigorously avoids hype and overstatement about the technology's capabilities.
Finally, let's talk about the integration of artificial intelligence, or AI. It seems AI is fundamentally changing the velocity of discovery in this field. How is it being integrated into this neuroediting renaissance?
AI and machine learning, ML, are the accelerators behind this speed. They are systematically analyzing the vast datasets generated in preclinical trials to predict optimal AAV vectors with high transduction efficiency. AI is also critical for optimizing guide RNA design for CRISPR systems, allowing researchers to computationally minimize those off-target risks we discussed.
So, AI helps solve the safety problem by increasing precision and identifying failure points much faster than human researchers could.
Exactly. This predictive capacity is what's driving the predicted mini-renaissance in neuroediting. It allows for the rapid personalization of therapy protocols based on an individual's genetic makeup and clinical needs. However, the governance of the AI itself is paramount.
And how does that governance interact with the need for safety and ethics?
Regulatory frameworks globally have to mandate stringent requirements for transparency, meaning the AI's decisions, especially for personalized dosing and vector selection, they must be explainable and auditable. We also must enforce comprehensive risk management and maintain essential human oversight to prevent reckless application. The sheer number of active cell and gene therapy applications just underscores the urgent need for efficient, AI-supported yet robust, human-governed regulatory processes to ensure this power isn't misused.
This deep dive has shown us the breathtaking capabilities emerging in molecular neuroscience. We have seen that the capacity to conquer debilitating diseases, to eliminate the crushing weight of trauma, addiction, and dementia, is rapidly advancing from theory to clinical feasibility. But the corresponding challenge to our identity and our social systems is equally profoundly present. The power to edit memories promises to eliminate pain, but it simultaneously asks us to fundamentally reconsider what it means to be an authentic human being defined by a continuous, coherent personal narrative.
The trajectory from this promising research to viable, widely accessible treatments depends critically on establishing trustworthy, adaptive, and globally coordinated governance that can actually keep pace with the science. This requires embedding continuous safety safeguards and proactive equity plans within national and international regulatory frameworks right now. We are at a threshold. The future of the self and the avoidance of profound social disparity hinges entirely on whether innovation can be governed by empathy and foresight, ensuring these powerful tools do not simply become luxury enhancements for a select few. The most important question is no longer can we edit memory, but should we? And if so, how do we protect the essential humanity of the people we are trying to save?